In this section, we're going to talk about how we take those electrical signals and translate them into mechanical contraction of skeletal muscle. So here's what we're going to look at. We're going to look at what a motor unit is, all the basic components, and how we recruit these motor units. We'll look at general muscle structure, we'll look at the macro as well as the microstructure, and we'll look at the functional characteristics of skeletal muscle. We'll look at the excitation-contraction coupling, the active state, the cross-bridge cycling, as well as the contractile properties of skeletal muscle. A motor unit is basically the nerve plus all of the skeletal muscle fibers that it attaches to. So there's a nerve cell body with an axon, there's going to be a neuromuscular junction where these two interface, and then of course we have the muscle fibers. If you look more closely at this diagram, you'll see this relationship between the nerve and the skeletal muscle fiber. I'd like you to pause the video and take a moment to look at all of the components of the neuromuscular junction. So start with box C. We have the synaptic cleft. This is basically a space that allows for a chemical messenger to be sent from the axon terminal to the motor end plate on the muscle fiber. So a motor nerve uses acetylcholine, or ACH, as a neurotransmitter. Those are the green balls that you see trapped inside the larger purple balls. These are what we call the synaptic vesicles, and they hold acetylcholine. The blue area is called the synaptic knob. When an electrical signal depolarizes the synaptic vesicles, the neurotransmitter acetylcholine is released and it travels across the synaptic cleft and binds with the receptors located on the motor end plate. The motor end plate then depolarizes and sends that signal across the skeletal muscle fiber. So we're going to talk about all these events in greater detail a little bit later when we talk about the events that lead up to a muscular contraction. As I mentioned earlier, a motor unit is comprised of the motor nerve plus all the skeletal muscle fibers that it innervates. In a muscle, we can have many motor units within that given muscle. As an example, in the quadriceps, we have hundreds of motor units that allow for varying levels of force production to be achieved. In our last lecture, I mentioned that nerves vary in terms of their speed. We have fast motor units as well as slow motor units. The fast motor units have a large soma or cell body and have a high resistance, meaning that it takes a large stimulus for that nerve to depolarize. They also have a short after hyperpolarization period, which means that we can stimulate them rapidly in succession. They also have a large axon diameter, which means that they can propagate the signal quite fast. All of this fancy terminology just means that the fast motor units are much harder to activate, their discharge rate is much higher, and their signal is transmitted faster. When we compare that with slow motor units, the soma or cell body has a low resistance, so it's much easier to activate these motor units. They have a long after hyperpolarization period, which means that you can't fire them one after another. There's a longer waiting period to get them to fire again. And they have a small axon diameter, which just means they can propagate the signal at a slower rate. So in other words, slow motor units are easier to activate, their discharge rate is lower, and the signal transmits slower. As I mentioned in the previous lecture, it's the nerve that's really going to determine the function of the skeletal muscle fiber. In other words, the characteristics that the skeletal muscle fiber has is dependent upon the nerve that is connected to it. So if you have a fast nerve attached to skeletal muscle fibers, those skeletal muscle fibers are going to have fast motor unit characteristics. Conversely, if you have slow nerves attached to skeletal muscle fibers, those skeletal muscle fibers will have the functions of a slow-acting motor unit. This brings us to motor unit heterogeneity. As exercise physiologists, we like to classify motor units into three categories: Type 1, Type 2A, and Type 2B, which is now known as Type 2X. This is used for edification purposes and is an oversimplification of reality. Human skeletal muscles actually have a variety of motor units, all with different characteristics, not just the slow to fast oxidative to glycolytic, which are the Type 1, Type 2A, and Type 2X respectively. We actually have a lot of different motor units in between these. This provides us with a functional continuum and allows us to have very fluid and smooth movements. If we only had three types of motor units, our movements would be far less fluid and very jerky. This slide shows you the staining of muscle fibers that is commonly used in research studies. As you can see, the variations in colors shows you how the continuum is actually very diverse and not just confined to those 3 types of motor units. With that said, understanding these 3 categories of motor units also allows us to understand the basic characteristics that each of these motor units might have and how the motor units in between might also behave. So in this table, we have our 3 basic motor units available to us. We have the type 1, which is also known as the slow oxidative, or SO for short. We have type 2A, which is known as the fast oxidative glycolytic. And we have the type 2B, or the type 2X, which is known as the fast glycolytic, or FG for short. On the left-hand side, we have a listing of various characteristics. The first on the list is predominant energy system. Does the muscle fiber use predominantly oxidative metabolism or glycolytic, or a combination of the two? What's the oxidative capacity of the muscle fiber? Is it more oxidative, less oxidative? What about the glycolytic activity? How much glycogen can that muscle fiber store? How fast can the muscle fiber contract? Is the fiber resistant to fatigue? What's the ATPase activity like? Remember, to change the rate of a chemical reaction, we have to change the enzymatic rate. And as you recall, ATPase is the enzyme that breaks apart ATP to release energy. What's the size of the fiber diameter? And how does that relate to force production? Myoglobin is the analog to hemoglobin, but in muscle. And remember, hemoglobin and myoglobin carry oxygen. How does that differ among the skeletal muscle fibers? And last on the list is how vascular these skeletal muscle fibers are. The more capillaries we have, the more blood we can bring into that skeletal muscle. A good question to ask yourself in order to remember these different types of muscle fibers is, what fiber types are these athletes using during their training? Is running different from weight training versus cross-country skiing? So let's answer this question. How does muscle contraction occur? First, let's take a look at basic skeletal muscle structure. We have the deep fascia that surrounds the entire skeletal muscle. The outer coating is called the epimysium. That surrounds all of the muscle bundles. The muscle bundles are surrounded by the perimysium. The muscle bundles then contain muscle fibers surrounded by the endomysium. The muscle fibers contain the myofibrils, which contain the sarcomeres, or the functional unit or the contractile elements of the muscle. You should also note that muscle fiber is equivalent to muscle cells, so you can either say muscle cell or muscle fiber. It's also important to note that the sarcolemma or sarcolemmal membrane is what surrounds all of the myofibrils. Once we get to muscle injury, you'll want to make note of what satellite cells are and where they're located. This is a closer look at a single myofibril. We're going to zoom in even closer and look at Z-disc to Z-disc. We're going to look at just the sarcomere to keep things simple, but just remember, sarcomeres are aligned end to end. This is the sarcomere. This is the functional unit of skeletal muscle fiber. Take a moment to study all of the components of the sarcomere. We have the thick filaments, which are the myosin filaments. These are where the myosin heads are located. And then we also have the thin filaments. It is the interaction between the thin and thick filaments that result in contraction of muscle. One of the proteins that keeps the thin and thick filaments together is titin. Titin is very important in keeping the thin and thick filaments right where they are so that the unit stays functional. These are scanning electron micrographs that show you the Z-disc to Z-disc and the thin and thick filaments. You can see that skeletal muscle fiber is very organized. So at rest, our thin and thick filaments might look something like this. But as we start to contract, we now have more overlap and interaction between the myosin and thin filaments. As contraction continues, you can see that thin filaments start to overlap, which means that force production is going to be reduced. Anyone who's ever weight trained has experienced the effect of these filaments overlapping. It essentially results in different force production depending upon the angle of the joint. So at some angles, we're able to produce less force compared with other angles. Think of a biceps curl. Where are we strongest in a biceps curl? Is force production going to be greatest when the biceps is in flexion or extension, or somewhere in the middle? In this diagram, what's important to note is that we have different structures in the muscle fiber or muscle cell that allow us to transmit the signal across the myofibrils. The sarcolemmal membrane is connected to the transverse tubules, also known as T-Tubules, and terminal cisternae. Those are those purple structures that you see in the picture here. That's then connected to the sarcoplasmic reticulum. It is this conduction system that allows the propagation of the action potentials across the myofibrils in a particular motor unit. So let's take a look at all the steps involved in getting skeletal muscles to contract. The electrical signal propagated along the axon reaches the synaptic vesicles at the synaptic knob and depolarizes these synaptic vesicles. That causes the synaptic vesicles to fuse with the synaptic knob membrane and then releases via exocytosis the acetylcholine into the synaptic cleft. This ACH then binds to ACH receptors located on the motor end plate. The binding of ACH to the ACH receptors depolarizes that motor end plate and causes depolarization to travel to the T-tubules. As the T-tubules are depolarized, this depolarizes the terminal cisternae of the sarcoplasmic reticulum. As the sarcoplasmic reticulum is depolarizing, it's releasing a very special mineral here, calcium. Calcium is then released into the sarcoplasm. The calcium binds to a protein called troponin, located on the actin filament. Once that calcium is bound to troponin, it allows tropomyosin, another protein, to slide off of the active sites of the actin filament. This exposes the active sites so that the myosin head can bind to the active sites. Prior to myosin binding to the active sites on the actin filament, we have to have ATP attached to the myosin head. Once ATP is bound to the myosin head is now free to bind to the active sites on the actin filament, so long as calcium is bound to the troponin protein that allows tropomyosin to expose those active sites. When action potentials are no longer reaching the motor end plate at the synaptic cleft, the calcium is taken up back into the sarcoplasmic reticulum. The enzyme responsible for this is sarcoplasmic reticulum ATPase. The rate of uptake back into the sarcoplasmic reticulum is of course going to be dependent upon the activity of sarcoplasmic reticulum ATPase. The higher the sarcoplasmic reticulum ATPase, the more calcium uptake. It's important to remember that myosin and actin filaments are 3D structures, and thus myosin heads and active sites are three-dimensional within muscle. This provides for an optimal arrangement to produce force. The shortening of sarcomeres, or the sliding filament mechanism as it's referred to, is dependent upon something called cross-bridge cycling. In the presence of calcium, the active site on actin is uncovered, as we have discussed, and the myosin head attaches. It swivels, it detaches, and returns to the original resting state. If calcium concentration remains high, the cycling continues. Many cycles are needed to move actin an appreciable distance. It's interesting to note that during a concentric contraction, only 50% of myosin is actually attached to the active sites on actin. In contrast, during an eccentric contraction, 70% of myosin is attached to the active sites on the actin filament. If all of the active sites on actin were occupied by myosin and shortened simultaneously, the muscle would only shorten about 1%. So in cross-bridge cycling, we continually have the attachment and detachment of the myosin head from the active sites on the actin filament. This is what allows muscle to shorten and produce lots of force. As you recall, ATP is energy, and it plays a very important role in this cross-bridge cycling. ATP is necessary to provide the energy for the power stroke and to dissociate the myosin head from the actin filament. So in other words, in order for the myosin head to attach to the active sites on the actin filament, you have to attach ATP before that attachment occurs. Likewise, in order to detach the myosin head from the active sites on the active filament after the power stroke is completed, we need to attach an ATP to release that myosin head from the active site on the actin filament. In the next few slides, we're going to talk about the genetic factors that affect force production. We're going to look at the frequency of stimulation, the number of motor units activated, the type of motor units activated, the size of the muscle's initial length when it's activated, the angle of the joint, as well as the velocity of shortening. With the frequency of stimulation, the more action potentials that you can generate in a given period of time, the greater the force production, as long as the muscle has not relaxed. So in the diagram, what you see in panel B and C is the summation of action potentials to produce a given level of force. So when you shorten the time between action potential arrival, you're going to increase force production. One of the reasons for this is that the passive elements, so tendons and ligaments, have already been stretched due to the previous stimulus. So as long as the muscle doesn't completely relax, the arrival of another action potential increases force. So here's another way to look at the summation of forces. The mechanical response to a single action potential is a muscle twitch. When an action potential is delivered to a muscle fiber before it's allowed to completely relax, the forces summate. In other words, they're additive. If the stimuli are close enough together, the maximum force potential of that fiber is reached. This is what we call muscle tetanus. As you recall, a motor unit is one motor neuron and all of the muscle fibers that it innervates. In general, motor units are recruited in order from the slowest to the fastest of motor units. In this example, motor unit 1 has a low threshold and is recruited for tasks that require low force development. As force requirements increase, more motor units are recruited to meet the increased force requirements. Each unit has a range of forces over which it responds. The force that each unit produces varies as the frequency of stimulation changes. The types of motor units that are activated also makes a difference in the amount of force that we can produce. In this diagram, we have the relative force on the y-axis, and on the x-axis we have time in milliseconds. We have a fast muscle, a mixed muscle, and a slow muscle represented here. The rates of force development and relaxation during a muscle twitch relate to the rate of cross-bridge cycling and the rate of calcium reuptake into the sarcoplasmic reticulum, respectively. Myosin ATPase and SR-ATPase of fast muscle have high activity rates, resulting in rapid force development and relaxation. Myosin and SR-ATPase in slow muscle have a much lower rate of activity, resulting in a slower rate of force development and relaxation. The size of the muscle is also going to make a difference. The bigger the muscle, the more force capability that muscle has. The muscle's initial length when activated is also a factor in force production. In this diagram, on the y-axis, we have the percent of maximum isometric tetanic tension. In other words, what's the percent of maximum force that can be produced? And on the x-axis, we have the percent of muscle length. In other words, how much interaction at the level of the sarcomere do the actin and myosin filaments have? At 80% of resting muscle length, we get 80% of maximum isometric tetanic force to be produced. In that diagram, you can see that there's a lot of overlap between the actin filaments. That blocks the active sites of the actin filament, so the myosin heads never get a chance to attach. That's why there's a lower force production at this given length. Conversely, at about 170% of resting muscle length, there's zero force production. Why? Because the actin and myosin filaments aren't in contact with each other. So there is no opportunity for the myosin head to attach to the active sites on the actin filament. At 100% of resting muscle length, or LO, as represented here by the dashed line, we have maximum force capabilities achieved. Why? Because there's an optimal interaction between the myosin heads and the actin filaments. So we know that muscles and connective tissue are elastic and they therefore store energy. If we stretch a muscle, we're going to increase the force production a little bit. So LO represents the genetically optimal length of a muscle where the force production is going to be the greatest. The angle of the joint also plays a role in force production. The force production is going to vary depending upon the angle that's created between the moment arm and the fulcrum. So throughout a joint's range of motion, you're going to get varying degrees of force production capabilities. The velocity of shortening also makes a difference in the amount of overall force that we can produce. On the y-axis, we have the percent maximum tetanic tension, so the percentage of maximum force that can be produced, and on the x-axis, we have the relative shortening velocity. During a concentric contraction, the force is going to be inversely related to velocity. So as the velocity is increased, the amount of force that we can produce is going to be less. Conversely, force production is greatest at lower velocities. During eccentric contractions, the amount of force that's produced is dependent on velocity only at the lower velocities. Once a certain velocity is achieved, it has no bearing on the amount of force that we can produce. So there is going to be an optimum speed of movement that's going to elicit the greatest power output. So the combination of force and velocity makes a difference in our power output, which is force over a given period of time. In the next few slides, we're going to cover the different types of muscular contractions. We'll be looking at static and dynamic contractions. An isometric contraction is where there is tension or force developed, but there's no shortening or lengthening of the sarcomeres. Training with isometric contractions leads to strength gains that are very specific to a particular joint angle. They're usually not directly applicable to most physical activity, therefore they're really not that useful. So as an example, if I took my hands and tried to pull my fingers apart after I've cupped them like this, I've got tension that's produced, but there's no shortening or lengthening of the sarcomeres. This makes me think about some of the isometric activities that I used to do in my soccer days. In particular, I recall putting my back up against a brick wall and having my knees bent at 90 degrees. And I had to hold that position for as long as I could. It was an isometric contraction. That really, in retrospect, had no practical application on the soccer field. If anything, it was more of a physical challenge and a mental challenge, but really didn't help any of the soccer players out on the field. Something to think about. In a concentric muscular contraction, the muscle force is going to be dependent on the joint angle, the muscle length, and the velocity of shortening. Whereas in an eccentric muscle contraction, the muscle force is going to be dependent upon the joint angle, muscle length, but is largely going to be independent of the velocity of shortening. In addition, an eccentric contraction is going to produce far more force per cross-sectional area than either an isometric or concentric contraction. If you recall, in an eccentric contraction, 70% of the myosin filaments are bound to the active sites on the actin filament. This results in greater force production per cross-sectional area. Therefore, I'm going to need fewer motor units to produce the same amount of force at any given load. I'm also going to use less ATP as a result of that. At submaximal loads, I'm also going to experience less fatigue. Isokinetic training requires the use of an isokinetic machine as the one you see in the lower right-hand corner of your screen. This machine allows us to overload the muscle through the entire range of motion at a constant angular velocity. This machine adjusts the resistance that a person encounters throughout the entire range of motion. So at the weaker joint angles, there's less resistance. At the stronger joint angles, there's more resistance. That helps us maintain that constant angular velocity. In research studies, it's been shown that the strength gains at the slower speeds are highly specific to velocities used in training. Conversely, with training at high speeds, there's an improvement in strength at both fast and slow speeds. That makes this machine very attractive for rehabilitation purposes. In plyometrics, also known as explosive jump training, we have a rapid eccentric contraction followed immediately by a concentric contraction. So how do we achieve this? In the picture over to the right, we see an athlete jumping off a wooden box down to the floor, and then what she's doing is immediately springing up. That achieves a plyometric. So what's going on here? As her feet contact the floor, that stretches tendons, ligaments, and muscles. This creates an elastic recoil. So when she flexes her calf muscles, there's already energy stored in those passive elements. That also results in the myotactic reflex being activated. So in combination with the elastic recoil and the stretch reflex, you can actually have an improvement in power output. And if we train in this manner, we can improve the power output over time. So with training, what we're trying to do is take advantage of this elastic recoil as well as the stretch reflex so that we can improve power output. So maybe the athlete pictured here is a volleyball player and she's trying to improve her ability to reach up high so she can spike the ball to the other side of the court. The problem with this kind of training is that you do run the risk of high musculoskeletal injuries just because of all the forces that are being applied very rapidly. One of the important notes to make about explosive jump training is that you don't have to have a high amount of weight to improve power. You only need about 8 to 10 reps 2 to 3 times a week. That's it. One of the rules of thumb that you want to make sure that you adhere to with this is that you should only increase the bench height gradually to increase the training effect. Increasing the frequency of training or the overall volume of work that you complete beyond the 8 to 10 reps 2 to 3 times a week is actually contraindicated. That's where you can increase the risk dramatically for those musculoskeletal injuries. On a final note, research studies suggest that if you stretch prior to any events that require these explosive movements, that you'll actually decrease your performance by doing so. So if you're a power lifter competing at an event, it would behoove you not to stretch prior to that event because you will actually decrease your performance. That could mean the difference between an athlete placing in an event versus not. One of the principles of training in exercise science that gets overlooked is that of training specificity. In this diagram, on the y-axis, we have the percent change in strength, and on the x-axis, we have the squat, leg press, and knee extension. Subjects in this study were tested for the squat, leg press, and knee extension strength, then trained for 8 weeks using squats only. Even though the same muscle group is used for all three movements, increases in strength with the squat were much greater than with non-specific leg press and knee extension. So in other words, the skeletal muscle should be conditioned in a manner similar to which they are to perform, including the exact pattern of movement. So think about it this way. If you wanted to get good at playing the guitar, would you practice the piano to get good at the guitar? Probably not. Same thing goes for athletes and their performance. If you want an athlete to improve at a particular sport or a particular function in sport, they have to practice that pattern of movement exactly during training. Otherwise, you're not going to get any increases in overall performance by doing activities that simply don't relate to that particular movement. Another training principle in exercise science is that of training overload. In order to increase muscle strength and muscular endurance, the muscle must be taxed beyond some critical level. So in other words, you have to stimulate the muscle beyond what it's normally used to throughout the day to get improvements in muscle structure as well as muscle function. We achieve training overload through the application of progressive resistance exercise, or PRE for short. Not only is PRE important, but we also need to allow ample rest between exercise bouts for muscle to recover and adapt. Too much exercise can lead to overtraining, which is going to hamper any adaptive response to that particular training and will eventually lead to overuse injuries. As a final point, the amount of improvement we get with exercise training is going to be inversely proportional to the initial fitness level. So in other words, if I'm an athlete that is well trained, the amount of exercise that I'm doing is going to give me far less than somebody who is sedentary. If I'm sedentary just starting out on a training program, I'm going to see much greater gains in my fitness level compared with the athlete. So this is basically the law of diminishing returns. If I'm already an athlete, doing more training isn't going to get me an improvement in my fitness level. But if I'm sedentary, then I've got a lot more to gain because I'm starting at a much lower point compared with an athlete.